Hello students, welcome to readmedprep Academy channel. Today in plant breeding part 3, we are going to talk about the milestones in plant breeding, green revolution and modern methods of plant breeding. Milestones in plant breeding, crop domestication was done initially. Farmers select the best wild species to create the crops. In 10,000 BC, domestication of wheat was done. In 1865, Mendel's laws were proposed. Gregor Mendel described the inheritance of traits from one generation to the next. His laws become the core of the classical genetics. Between 1865 and 1926, plant breeding was done based on crossbreeding, development of improved varieties by combining good characteristics from two parents. In 1926, hybrid breeding was done, crossing two genetically different individuals to develop better performing hybrid which has more vigorous hybrid corn. Example, more vigorous hybrid corn was produced. In 1940, blast resistant rice was done. Mutagenesis was done, developing new genetic diversity by exposing the crop plants to chemical agents or radiation. In 1953, understanding of the structure of the DNA, James Watson and Francis Crick proposed the double helix structure of the DNA. In between 1953 and 1994, plant breeding was done based on the genetic information, development of improved varieties by working directly with the DNA. In 1994, genetically modified organisms were introduced, introducing foreign genes into the DNA of a plant. For example, insect-resistant cotton was produced. In 2000, barely resistant to yellow dwarf virus were produced. Marker-assisted selection was done, locating the desirable traits in a plant for efficient selection and breeding. Now, targeted breeding is done using modern tools such as genome editing for more targeted breeding. And the future is very good in plant breeding. To sum up, for 10,000 years, farmers and breeders have been developing and improving crops. For 150 years, plant scientists and breeders have improved plant breeding on a scientific basis. Today, farmers feed at least 10 times more people using almost same amount of lands as 100 years ago. By 2050, we will need 50% more food to feed the rapidly growing population. Now, what is green revolution? Green revolution is the term that was coined by William S. Gould in 1968. It is defined as the cumulative result of a series of research, development, innovation and technology transfer initiatives. Agricultural production, especially wheat and rice, manifolds worldwide, particularly in the developing countries between the 1940s and the late 1960s. Green revolution or the third agricultural revolution is the intensive plan of 1960s to increase the crop yield in developing countries by introducing the high yielding resistant varieties, increased irrigation facilities, fertilizer application and better agricultural management. The scheme began in Mexico in 1940s and was successfully introduced in the parts of India, Asia, Middle East and Latin America. Dr. B.P. Pal, the director of Indian Agricultural Research Institute, requested M.S. Swaminathan to arrange for Dr. N.E. Borlaug visit to India and for obtaining a wide range of dwarf wheat possessing the Norin 10 dwarfing genes from Mexico. In 1963, semi-dwarf wheat of Mexico was introduced, from which India got 5 prolonged strategies for breeding a wide range of high varieties like Sonora 64, Sona Laika and Kalyan Sona, possessing a broad spectrum of resistance to major biotic and abiotic conditions. Same as wheat, M.S. Swaminathan produced the first semi-dwarf fertilizer responsive hybrid variety of rice TN1, Taichung Native 1 in 1956 from Taiwan. The derivatives were introduced in 1966. Later, better yielding semi-dwarf varieties of rice, Jaya and Ratna, developed in India. The dawn of green revolution occurred in India. Norin 10, the cultivars found that Norin 10 dwarfing genes have high photosynthetic rate per unit leaf area and increased respiratory activity. You can see in the image on the right side, the fulls or glassy fulls and Daruma were crossed in 1917 to produce fulls Daruma. Fulls Daruma and Turkey red wheat were crossed in 1924 to produce Norin 10 wheat variety. Gonjiro Inazuka of Japan selected the semi-dwarf wheat variety that became Norin 10. He would have never thought that the semi-dwarf genes would not only revolutionize the world of wheat, but also help to save more than 1 million lives from hunger and starvation. Plant breeding was done for developing resistance to diseases. Some crop varieties bred by hybridization and selection for disease resistance to fungi, bacteria and viral diseases are released. Here in this image, you can see the table where the crop on the left side, variety in the middle and resistant to diseases on the right side. Wheat, the variety is Himgiri, and resistance to diseases like leaf and stripe rust and hill bunt was produced. Brassica, Pusa swarnim, otherwise called Kararai, variety was produced which was resistant to white rust. Cauliflower, Pusa shubra, Pusa snowball, K1. These varieties were resistant to diseases like black rot and curl blight, black rot. Cowpea, Pusa komal variety was produced which was resistant to bacterial blight. Chili, Pusa and Sadabahar. The varieties were resistant to diseases like chili mosaic virus, tobacco mosaic virus and leaf curl. Now let us move on to the plant breeders. Norman E. Borlaug, the plant pathologist and the plant breeder, devoted his life at the International Maize and Wheat Improvement Center at Sonat in Mexico. He developed a new high-yielding, rust-resistant, non-lodging dwarf wheat varieties like Norin 10, Sonora 64, Lerma Rojo 64, etc., which are now being cultivated in many countries. This formed the base for Green Revolution. He was awarded a Nobel Prize for Peace in 1970. Dr. M.S. Swaminathan, he is the pioneer mutation breeder. He has produced Sharbati Sonora, is the amber grain colored variety of wheat by mutation, which is responsible for the Green Revolution in India. Dr. Swaminathan is called the father of Green Revolution in India. Nel Jayaraman. Mr. Jayaraman hails from Adirangam village in Tiruvarur district. He was a disciple of Dr. Namalwar and state coordinator of Save Our Rice campaign Tamil Nadu. He strived hard for conservation of traditional rice varieties. He had trained a team of farmers and regularly updated them on the current issues that affect them. In 2005, he organized the first ever traditional paddy seed festival in his farm as an individual. The seed festival in May 2016 at Adirangam was the 10th in a row and in which 156 different traditional varieties were distributed to more than 7,000 farmers across Tamil Nadu. He was invited by the Philippines government to give a talk at the International Rice Research Institute IRRI on his work and mission. In 2011, he received the state award for the best organic farmer for his contribution to organic farming, and in the year 2015, he received the national award for the best genome savior. Now, what is biofortification? Breeding crops with higher levels of vitamins and minerals or higher protein and healthier fats is the most practical means to improve the public health. Here, biofortification you can see can be done by traditional biofortification, where the growth of the plants are done to increase the level of nutrients. Biofortification is done by breeding, by selective breeding, which helps in the plants to produce high levels of nutrients. By transgenic biofortification, where the plants are taken, new genes are introduced into the plant for increasing the level of nutrients. Breeding for improved nutritional qualities undertaken with the objectives of improving the protein content and quality, oil content and quality, vitamin content, micronutrient and mineral nutrient. In 2000, maize hybrids that had twice the amount of amino acids, lysine and tryptophan compared to the existing maize hybrids were developed. Wheat variety Atlas 66, having a high protein content, has been used as a donor for improving cultivated wheat. It has been possible to develop an iron fortified rice variety containing over 5 times as much as iron in commonly consumed varieties. The Indian Agricultural Research Institute, New Delhi, has also released several vegetable crops that are rich in vitamins and minerals. Example, vitamin A enriched carrots, spinach, pumpkin, vitamin C enriched bitter gourd, patua, mustard, tomato, iron and calcium enriched spinach and batura, and protein enriched beans, broad beans, lab, French and garden peas. Here you can see the fortified plants. Sweet potato was fortified with vitamin A, cassava with vitamin A, maize with vitamin A and zinc, banana or plantain fortified with vitamin A, beans fortified with iron, pearl millet fortified with iron, cowpea fortified with iron and zinc, Irish potato fortified with iron and zinc, sorghum fortified with iron and zinc, lentil fortified with iron and zinc, wheat and rice fortified with zinc. Now sugarcane, Saccharum barreri, was originally grown in North India but had poor sugar content and yield. Tropical canes grown in South India, Saccharum officinarum, had thicker stems and higher sugar content but did not grow well in North India. So these two species were successfully crossed to get sugarcane varieties, combining the desirable qualities of high yield, thick stems, high sugar content and ability to grow in the sugarcane areas in North India. Resistance to yellow mosaic virus in bhendi, Abelmoschus esculentus, was transferred from a wild species and resulted in a new variety of Abelmoschus esculentus called Parbharni Kranti. Plant breeding was done for developing resistance to insect pests. Insect resistance in host crop plants may be due to morphological, biochemical or physiological characteristics. Hairy leaves in several plants are associated with resistance to insect pests. Example, resistance to jassids in cotton and cereal leaf beetle in wheat. In wheat, solid stems lead to non-preference by the stem, sawfly, and smooth leaves and nectarless cotton varieties do not attract bollworms. High aspartic acid and low nitrogen and sugar content in maize leads to resistance to maize stem borers. Here you can see the pest resistance varieties, crop on the left side, variety in the center and insect pest. Brassica, called rapeseed mustard, the variety is Pusa gaurav, was produced with insect resistance to aphids. Flat bean, Pusa sem 2, Pusa sem 3, which is resistant to jassids, aphids and fruit borer. Okra, bhindi, Pusa savani and Pusa A4. It is resistant to shoot and fruit borer. Now, what is modern plant breeding? In the milestones of plant breeding methods, genetic engineering, plant tissue culture, protoplasmic fusion or somatic hybridization, molecular marking and DNA fingerprinting are some of the modern plant breeding tools used to improve the crop varieties. New plant engineering techniques or new breeding techniques (NBT) are a collection of methods that could increase and accelerate the development of new traits in the plant breeding. These techniques often involve genome editing to modify the DNA at specific locations within the plants to produce new traits in the crop plants. The various methods of achieving these changes in traits include cutting and modifying the genome during the repair process by tools like CRISPR or Cas. Genome editing to introduce changes in a few base pairs using a technique called oligonucleotide directed mutagenesis (ODM). Transferring a gene from an identical or closely related species called cisgenesis. Organizing processes that alter the gene activity without altering the DNA itself, epigenetic methods. Now, let us see this image where the new generation plant breeding is done. The traditional methods, crops with improved characteristics produced by cross-breeding plants and desired genes. The gene has 50-50 chance of being passed to the offspring. Desired gene will take multiple generations to spread. Modern methods combines genes with different species. The bacterial chromosome, which is a plasmid or a circular DNA molecule, is removed from the cell. This acts as a vector to carry the gene. The DNA containing gene for desired trait is removed from the chromosome and restriction enzyme cuts the gene from the DNA. Then it is used as a vector. The restriction enzyme cleaves the plasmid. Second enzyme, called the DNA ligase, pastes the gene into the DNA molecule, making it a recombinant DNA vector. And this vector is inserted into the plant cells and a new plant is obtained with the new characteristics. Now, what are the new breeding techniques? Plant with desired gene has gene pasted into all its chromosomes. Gene is transmitted to nearly all offsprings. CRISPR or Cas9, which is a precise gene editing, cuts the DNA at specific sequence and enables introduction of replacement sequence. Clustered, regularly interspaced short palindromic repeats is the expansion for CRISPR, and Cas9 is a cleaving protein. So today in plant breeding part 3, we discussed about the milestones in plant breeding, green revolution, modern methods of plant breeding. So thank you. Kindly subscribe, like, share and comment to channel Read MedPrep Academy. Register in our website for UG and PG MCQs www.readmedprepacademy.com. Our Facebook ID is Read MedPrep Academy. Our email is readmedprepacademy@gmail.com. Our Instagram is Read MedPrep Academy. Kindly post your questions in the comment box. We will reply with appropriate answers. Join Read MedPrep Academy for UG and PG NEET MCQ preparation for 2022 and 2023. Kindly WhatsApp the number given below. Thank you very much.